Good morning. I'd like to welcome all of you to my course on modern methods of brain exploration with a focus on functional magnetic resonance imaging. And I really appreciated the welcome this morning. That was very nice. And I'm glad to be here with all of you. Mihir already gave you a bit about my background, but I just wanted to go through it briefly again and to point out that the way that I got to functional neuroimaging, which is my current field, was a little bit of a crooked path. I began in the Department of Engineering, Science, and Mechanics at the University of Tennessee, and I knew that I enjoyed the mathematics and the quantitative sciences, but I also enjoyed biology as well. My parents, my father was an engineer and my mother was a nurse, and I thought it would be nice to do something in the middle of the two. So I ended up in the engineering, science, and mechanics with a focus in biomedical engineering. And at this place, I did a lot of biomechanics, fluid mechanics, modeling blood flow, and things like that. But I knew I wanted to study the brain. That was my area of interest that I was passionate of. So when I started at the University of Pittsburgh, I moved into the Department of Neurology and did research there. We studied heart rate variability in sudden infant death syndrome, which is SIDS. I don't know if you probably have some of that here, but we were looking at how heart rate variability was different in children who are at higher risks for SIDS. And I also did some neurosignal processing and prediction by looking at data from the neuro ICU unit. The patients in the neuro ICU unit are often unconscious and unable to communicate because of their severe head injuries, but they're constantly monitored with all kinds of signal collection. And so we collected those signals and tried to create models in order to predict if the patient was going to make a turn for the worse, or so we could know ahead of time if they needed extra care or extra treatment. So in doing so, I sort of got into the signal processing of the neurological system, and I moved on to a postdoctoral fellowship in child and adolescent psychiatry. And at this point, I was fortunate enough to start studying functional MRI and moving from the two-dimensional sort of signal processing mode to the three-dimensional mode of image processing. Afterwards, after I finished my PhD, I worked in the Department of Pharmaceutical Sciences and Radiology and Bioengineering at the University of Pittsburgh, and currently I'm a senior research fellow at the University of Pittsburgh Medical Center, where my main area of focus is functional MRI. The course I'm going to be teaching to you is sort of new for me to teach from start to finish. I've given pieces and parts of this lecture, but never from start to finish. First, we're going to go over a brief introduction to neuroscience, including a little bit of history on the topic, how they used to study the brain in the olden days before they had imaging techniques to do so. And as a background to this, we're going to go over an introduction to the nervous system, how the nervous system communicates throughout the body, how the brain sends signals and receives information. And we're going to go over a little bit of neuroanatomy of the brain. Then our next area of focus is going to be talking about mapping function to structure. So when you talk about anatomy, you're really talking about the structure, but when you talk about function and mapping function to structure, you're asking the question of where in the brain do different things live? Where do emotions live in the brain? Where do thoughts live in the brain? How is your behavior controlled, and where is that happening in the brain? So we're going to get into that, and from that we're going to get into the anatomical and functional neuroimaging techniques, which is really, these techniques have allowed the study of the brain to advance quite significantly in the past probably 30 years or so. We're going to talk a lot about magnetic resonance imaging and then get into really the meat and potatoes, the functional magnetic resonance imaging. So we're going to talk about the bold signal, designing experiments, how we model the data, some statistical problems that we face. We're going to do a little mini lab session and talk about safety. And then we're going to get into some newer techniques. One is functional connectivity, and resting state fMRI, and another is diffusion tensor imaging. Both of those techniques use MRI in different ways to ask different questions and address different questions about the nervous system. And finally, we're going to get into specific applications. So some of the lectures that I have are longer than others, so we're just going to keep going. I know today we have a little bit of a shorter day, but if we end a little, if one lecture takes a little bit shorter than another, we're just going to keep going on so that I try to have enough time to get the whole course in and talk about the applications that I'm interested in. So the main ones that I'm going to plan on talking about are sleep and memory and learning. Okay, so I know all of you come from different backgrounds. Well, I'm assuming all of you come from different backgrounds. So I'm going to try to keep this course as conceptual as possible, just to try to give overviews and give a general idea of the basic concepts without a lot of the hard, complex background that goes into them. So if you're uncomfortable with equations and things like that, you may see a few of them in here, but don't worry. Don't be scared and feel like you're falling behind. So if you don't have a deep understanding of it, it's okay. I want you to just gain a basic understanding of the brain and the nervous system. I want you to get a basic understanding of imaging techniques, especially MRI and functional MRI, and a basic understanding of how these techniques are extended to use in different types of MRIs, such as diffusion MRI and resting state, and some applications of these techniques. So I want to note here that please don't be afraid to ask questions, or if I'm speaking too quickly, or if you can't understand something I'm saying, please raise your hand, tell me to slow down, and tell me to go back over it. I know I probably talk a little bit more quickly when I'm nervous. So am I okay? Are you guys all, am I speaking too quickly? Okay, just tell me to slow down if I am. I will not be offended at all. And we're going to have a brief quiz at the end, but it's not going to be anything difficult, so don't feel worried about that. I know in the US, we all often start by telling people to turn their cell phones down to vibrate. And so if you could all do that now. And this is mostly for my benefit. I know when I'm nervous and a noise comes up, sometimes I jump out of my skin. So I don't want to be jumpy up here. But if you do need to talk, just feel free to go out. And, you know, I know people have lives and things going on. So I won't feel offended if you need to take a call and walk out. All right. All right. So before I start on all the scientific things, I just want to go over why I was so interested in studying the brain. So I'd like to propose that the brain is probably one of the greatest mysteries of our time. And this, of course, is my opinion. I know there are many mysteries in the universe, but the reason that I think this is because our brain is thought to be responsible for our thoughts, our perceptions, our behaviors. Our brain takes in constantly sensory information from all around us. It integrates this and creates the model world that we see and interact with every day. Our brain is responsible for our movements, our balance, our coordination. Our brain is responsible for storing and retrieving memories, for learning and adapting to our environments. It's responsible for our emotions and our fears. These things often help us in our environment to be cautious of things and feel a different way about things and keep us safe. And our brain is also responsible for sleeping and resting of the body. So all of these things, but the thing that I think is the most interesting is that we really have very little understanding of how the brain works. So it does all these things for us, and yet we still don't really understand how it works, which I think is just fascinating. And also probably a little bit good, because if we could get our fingers in there and turn the dials and the buttons, surely we would probably mess something up. So maybe it's better that we don't fully understand how it works so that we can't try to adjust ourselves and mess something up. But Anyway, so I hope you all find the brain as interesting as I do. So we're going to start out with some perception experiments, and you might have already seen some of these, so just bear with me if you have. Okay, so the first one is Adelson's same color illusion perception. And these are designed to sort of show us that our brain is really perceiving what we're taking in as sensory information. It's not necessarily seeing what is there in front of us. It's the way that we're interpreting information. So look at the drawing below. Which square do you think is darker? Square A or square B? Square A, right? Square A definitely looks darker. But in fact, if you draw a line between the two, you see that they're actually the same color. So this is actually a perceptual illusion of the brain. And even after looking, it's sort of hard to believe, isn't it? But they do say that they're the same color. So even when we know the colors are the same, they still appear to be different. So we expect the cylinder to cast a shadow, and we expect square A to be darker because of the pattern of the squares here. So in this case, our brain is actually seeing something different from our eyes, and that's, again, the perception image that our brain is using. So in this picture, which monster is larger? Is it the one in the front or the one in the back? Or are they the same size? Well, I think the one in the back certainly looks bigger, but in fact, they're actually the same size. So our brain is interpreting these depth cues of the tunnel here to make it appear that the image in the back is larger when in fact it's not. Okay, here's another one. So if you can count the black squares between this black dots between the squares in this picture, then you'd be doing pretty well. So if you try to focus on the dots inside, they're actually white. But as you see in the periphery, all these black dots around it. So this is another illusion that how our brain is perceiving these. There are no black dots in that. Our visual system works a lot on processing edges of objects, and edges help to define and understand an image in our brain. So these visual artifacts or illusions occur when we look away from the center of the receptive field, and in fact they really are artifacts or illusions that our brain is perceiving as we try to focus. Okay, so I think this is no, we have one more after this. So here's a hidden animal picture. So in the first one on the left, do you see a horse or do you see a frog? I think when I first look at it, I see a horse, but I can see the frog if I sort of look at it a different way. The one on the right, do you see a rabbit or do you see a duck? I think when I first look at it, I see a rabbit, but I can also see the duck. Can you see both at the same time? Some people can, I guess. And some you sort of have to switch your brain either way to see one or the other. Again, this is how our brain is perceiving things and making sense of what's in front of us. Okay, I think this is actually the last one. The lines here are actually parallel, but they don't look parallel to me. Maybe some of you think they look parallel, but because these squares are slightly offset, it makes it look like the lines are sloped. So again, our brain has expectations of how we see the lines and how we expect them to flow, and so those slight offsets make them look like the lines are sloped. Okay, so perception. Perception is perceiving this 3D world in spite of only seeing a two-dimensional picture in both eyes. So our retina is actually two-dimensional, but we see the world in 3D. And part of this is because of the relative position difference between each of our eyes. So the difference that's between our eyes is a cue for the brain to help it understand how things are at a distance from us, and helps us to make this 3D image out of the two-dimensional image. And as I mentioned before, the brain is constantly taking in sensory information and constructing our perception of the world around us. So what you're actually seeing and taking in is a model that your brain is producing to help you understand what the world is around us. And we'll see a little bit more of this as we go through the course. So I wanted just to start with some human facts of the brain by the numbers. The typical brain is about 2% of the body weight, but it uses 20% of the total energy and oxygen of the body. So it's actually a very important organ. Your body puts a lot of energy and oxygen into it. The brain is 73% water, but it only takes about 2% hydration of your body for your attention, your memory, and your other skills to start being effective by this dehydration. The brain weighs about 3 pounds, but of that, the dry weight of the brain is 60% fat, making your brain the fattiest organ in the body. And the reason for this is that we'll learn in a little while, because of the white matter in the brain is wrapped in these myelin sheaths, which is actually fatty tissue. And that is a very important element in the brain that helps the brain process this fatty tissue, and we'll talk about that a little later. No one knows an exact count, but the latest estimate is that our brain contains roughly 86 billion neurons. So that's a lot of cells, brain cells, and neurons are the brain cells that help communicate in the brain and throughout the body. We'll also talk about them a little bit more. Each neuron is thought to contain on average about 40,000 synapses. Synapses are the connections between the brain cells, between the neurons, and between different parts of our body. So each cell is widely connected to other parts of the body and other parts of the brain. A piece of brain tissue the size of a grain of sand contains about 100,000 neurons and about 1 billion synapses, all communicating with each other. So if you really stop and think about it, it's a very, very complex system. The number of studies on the brain have advanced greatly with advancements in methodology, particularly in anatomical and functional neuroimaging techniques. So here's a little plot that I found in a journal paper that shows the amount of brain research over the past from 1990 to 2010. And you could see in 1990 there were a very limited number of brain studies, and some of these techniques we're going to talk about that they have there on the, I guess I would call that the z-axis. fMRI, other methods, EEG, behavioral studies, because now where it wasn't possible before, it's now possible to study the living, functioning brain in a non-invasive manner. And you're going to hear the word non-invasive a few times during this course. Non-invasive means that we don't have to cut it open. We don't have to inject things into it. We can study the living brain in people without having to subject them to any sort of nasty medical techniques, which is, I find it extremely fascinating. In fact, I tell one of my sons that we can actually study the brain and take pictures of it in the dark inside the head without actually having to take it out. And I just think that is quite fascinating, and one reason why I got into this field. Now we're going to go over a brief history of neuroscience, and by brief I mean pretty brief. But what I thought was interesting, that as far back as 1700 BC, there was evidence that people actually knew about the nervous system and studied into the nervous system. And there's evidence in 600 BC of an Indian physician who describes the nervous system and nervous system diseases in a manual that he wrote. I'm not going to try to pronounce it because I'm afraid I'll make a fool of myself. Hippocrates, around 460 to 379 BC, discussed epilepsy as a disturbance of the brain, which was very modern for that time, because people thought that epilepsy was some kind of a spiritual or infliction disease that had to do with the devil. And interestingly, some parts of the world, they still believe that, so, which I find really interesting. But as far back as then, he knew that it was a disturbance of the brain. Hippocrates and Plato were some of the first of the more, I guess, modern times to say that the brain is actually the seat of the mental process. So the brain is where we think. Aristotle believed that the heart was the seat of the mental process. He thought that we think with our hearts and that our brain was sort of a cooling system for the heart, which now we know that was incorrect. Well, I was looking through brain history. I found this very interesting thing on the internet about the world's oldest brain surgery, and it took place here in India, so I thought I would show you a picture of it and bring it up. It's estimated to be 43,000 years old, of this skull. And what's so interesting about it is that by examining the skull, they saw that this person, you can see on this right picture where the arrow is, top right, that this person actually had some sort of an injury to their skull. And when you get a brain injury, oftentimes what happens is the brain swells and the tissue swells inside the head, and the pressure gets to be too high. And nowadays they do often do surgery by drilling holes in the head to relieve the pressure. And interestingly enough, that's what they did 43,000 years ago in this case. They drilled some holes in this head to remove some of the damaged, shattered bones and the pressure that's built up in the head and the pools of blood. And in fact, in this skull, not only was the injury present, but there was also evidence of healing after the injury. So they called this the first successful brain surgery, and so long ago, and techniques that we still use today. Okay, now we're getting to the age of Enlightenment, 1700 to 1800s. And at this time, I'm focusing on a little bit because this is the first time that we know of in written history where people were thinking, where is the actual mind? Where does the mind live? Where is the function related to the structure of the brain? So they knew by this time that the nervous system was electrical in nature. And there was a man named Franz Joseph Gall and a student of his that got into this field called phrenology. I don't know if any of you have ever heard of it before, but it was very interesting to me because they were the first ones that tried to localize brain function to the structure underneath of the brain. So what they did was they anatomically divided the brain into 35 functional brain organs. And if you can see here, they actually parcellated the brain up into these 35 different sections. And they said that each section was responsible for some sort of intellectual or emotional faculty that a person had, a part of their personality or some trait that they could attribute to each of these sections. And what they thought was that the shape of the brain underneath was equal to the shape of the skull. So they thought if you had a larger lump in one area of the head, then you had more of that attribute than another person had. And just to give you an idea of some of these 35 regions that they attributed, they thought one area was involved with physical love, one area was involved with parental love, which is the particular feeling which watches over and provides helpless offspring, or parental love. So if you had a big lump in that area, you are more inclined to give parental love than another person. And you can see, combativeness, which is your person's disposition to quarrel and fight. Destructiveness, which, somebody's disposition to destroy things. And you can read the others here for yourself. But it was sort of the first, like I said, the first time that we have evidence that people were actually trying to attribute different qualities of a person to the actual brain itself in different areas. So if you see here, they call this the actual only true science of the mind, and now we look back and think maybe it was a little silly, but it certainly was one of the steps towards trying to understand the functioning of the brain. Okay, functional localization. So this is the mapping of brain's function to brain structure. And like I said, at the age of Enlightenment they started thinking about this question and the question of functional localization a little more intently. And there were two examples of two different physicians at this time. One was named Dr. Broca. He noticed that a stroke patient that he treated had lesions in a certain part of the brain, and this stroke patient could not speak properly. So he had spontaneous speech and motor speech control problems. But he did not lack the understanding of the spoken language. So you could speak to him and he could understand what you were saying, and he knew what he wanted to say. He just couldn't get the speech out. And the same sort of problem with Dr. Wernicke. He also had a stroke patient. These two guys were operating independently. This guy could still speak with connected words, but he lacked the ability to understand the meaning of the spoken words and sentences. So after these two patients died, they opened up the head and looked to see where these areas were. And nowadays, we have actual areas attributed to each of these physicians. There's Broca's area and Wernicke's area. You can see them labeled here. And what we call nowadays, that these patients suffered from, is called aphasia. So this aphasia is the difficulty, language disability or difficulty caused by a brain injury. So if you have a damage to Broca's area, you will have Broca's aphasia. If you have damage to Wernicke's area, you might have Wernicke's aphasia. And so we still have these areas today, and we still attribute these functional areas to a structural location in the brain where this type of processing takes place. I thought it would be interesting just to see an example of two different patients with these types of disorders. So if this works, maybe not. We'll be able to see an example of each. All right, so maybe if I try from here.
Stroke. Stroke. 5 years.
5 years.
Yeah. 5 years. Drive car, okay? Me. Drive car.
All right, I'm going to ask you to tell me some answers to these questions. What do you do with a hammer?
Found. Yes.
You write with it?
No.
You eat with it?
No.
You pound with it?
Yes.
Okay.
So he does know the words, he just can't make the sounds of it. So, all right, let's watch the second one, which is the Wernicke's aphasia. So you can see he speaks pretty fluently, but his...
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people over them over there. they're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck, for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here, and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
With the iPad that we were doing. Like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them.
Good.
This girl was very good and happy. And I play golf and hit other trees. We play out with the hands. We save a lot of hands on hold for us, other hands. I don't know what you get, but I talk with a lot of hand for him. Sometime. Am I talk of any more to saying?
All right. Thank you very much.
Thank you very much. I appreciate it, and I hope the world lasts for you.
Thank you. It's been a pleasure. Bye-bye.
Have a good day.
So you can see he's sort of a happy kind of guy. The interesting thing about Wernicke's aphasia is that I don't think people often realize that they are not using the appropriate words. A good friend of mine, her mother unfortunately had a brain tumor, and she had it in the temporal region, which is where these language areas are, and that were some of the early signs that my friend noticed, that her mother would text her, but she would use the wrong words for things, and that was like a very, very early manifestation of the tumor. So, okay. So those were two examples of aphasia and how an injury in a very specific anatomical region of the brain can lead to a functional deficit in a person. Another person who tried to do functional segmentation of the brain pretty early on was this man called Brodmann. He divided the brain into 52 distinct functional areas, and he did this based on a brain dissection and very intricately looking at different types of tissue throughout the brain. And based on these, where the tissues were different, he divided these into functional areas. And the very interesting thing about Brodmann areas are that we still actually use them today in our brain imaging techniques. So he did a pretty good job when he did this, although some of the functional areas we're not really specifically sure. We're learning a little bit more about what each functional area does through more research. So we've touched a little bit on this when we talked about the stroke patients and opening up their heads afterwards to see sort of what the deficit was. But as we go through this history, I just wanted to point out how some of the people early on studied the brain before imaging techniques came along. So one method was to study animal models, and a lot of people still do this, of course. You don't have one advantage, of course, is that you don't have to use live humans, but there are several disadvantages. One is that there are limitations on functional experiments. So if you really want to study human function, it's difficult to translate these animal studies to actual humans. You can't really ask an animal to tap his finger or to say some sort of word while you're studying him. So you have limitations on what you can actually do. And like I said, their brains are different, and sometimes this doesn't translate well to human brains, but it's still, to this day, a very effective method of trying to understand some of the areas of and how the brain works and how different cells work. Another method early on that people used was this postmortem dissection. And you can, of course, know that the limitations, the advantages to this are that you don't have to use live people, but samples are, of course, limited. You have to have somebody's permission nowadays to cut their head open and take a look at their brain. And, of course, you can't do functional experiments on a dead brain. So that's a definite limitation of trying to understand functional organization in the brain. Like the methods I talked about with Broca and Wernicke, another method to study the brain was to wait simply until someone had a disease or an accident, or some kind of a surgery or a brain problem, and then sort of look at what their loss of function is and attribute this function and the loss of it to the injury or the brain lesion or the site of the tumor or stroke occurred. And a guy named Angelo Masso, he actually used people who had skull deficiencies, and you could see into their brain, and he noticed that when they were doing different things, the distribution of blood flow during these tasks changed by looking at the pressure waves in the head. And this is actually, in fact, now the basis of functional MRI, understanding where these blood flow changes in reference to different functional tasks that people do. So this was an interesting thing, but of course he had to wait for somebody with a skull deficiency to come along before he could study that. So these types of studies were indeed studies of opportunity only. So one of the very famous studies of opportunity was this man called Phineas Gage, and I'm sure if you have studied anything about the brain, you've probably heard about this man before. He was a 25-year-old railroad worker, and he was actually a foreman on the railroad, and he was working with a blasting crew when there was an accident that drove this big iron rod right through his head. So the rod entered through his cheekbone, you can see in the picture, and passed his eye and out the top of his head. And interestingly enough, he survived this trauma, but people noticed a lot of changes in Mr. Gage after he had this accident. So before the accident, he was very skillful and well-liked. He was described as the most efficient and capable foreman of the railroad working. And after this injury, a big part of his left frontal lobe was destroyed. And people noticed these big changes in his personality and his behavior. So whereas before he was skillful and well-liked, after the accident he became very fitful, irreverent, profane. So he sort of lacked this sort of behavioral control that allowed him to monitor his behavior and act appropriately in a social way. So they described him as no longer Gage. And I imagine if any of us had some sort of accident of that scale, maybe we'd be pretty crabby about it too. But these were indeed changes in his personality and behavior that were attributed to a very specific localization point in the head. And so they deduced from Mr. Gage that the frontal part of the brain was responsible for the more complex functions of decision making and social cognition, sort of controlling how you act in a social environment and your behavior to others. Okay, so you could see that the technology was very limited in the early studies of brain, but as the 1900s rolled around, there were a lot of new methods that came about, fortunately for neuroscience research, that allowed us to study the brain better. Around 1895, where the first X-rays were discovered. EEG was first measured from the brain, and this is electrical activity, in 1924 in Germany. Then skip many years ahead to the 1970s, and people started using computer-based brain scans, CAT scans and CT scans, we know of them as. PET imaging, which is positron emission tomography, came about in about 1973. This is a method used to measure metabolic functions. Magnetoencephalography is similar to electroencephalography, but instead of measuring electrical waves from the brain, we're measuring magnetic activity from the brain. And of course, in the 70s is when they began using magnetic resonance imaging. And whereas X-rays and CAT scans were very limited in the ability to see soft tissue, MRIs came out and you could see beautiful images of soft tissue using this technique. And of course, in the 1990s, functional MRI came about, and since then, these types of studies have just exploded in the field of psychology and psychiatry. We're going to talk a lot about each of these different methods in the upcoming days. But before we get started on those, I'm just going to give you some background. Our next lecture is going to be some background on action potentials and how the brain communicates with how cells communicate with each other and how the brain communicates with the body. Lecture 3, we're going to talk about the brain lobes and basic neuroanatomy of the brain. And in lecture 4, then we're going to start talking about the actual functional systems of the brain, in which areas of the brain work together to allow us to do different things like process sensory information, control our emotions, use our memory processes and things like that. So those will be the things that we'll be talking about in the days to come. So that ends my first lecture. Do we want to take a small break and then do the second? Maybe 15 minutes? Yeah, perfect. Okay, thank you. Yeah, please ask questions if anybody has any. Okay, just a few minutes and then we'll start on lecture 2. Okay. So now we're going to talk about the nervous system. Can you see me better? Okay. I want to make sure I can use the microphone too. All right, so this is just a brief introduction to the nervous system, just to have a background of the things that we're going to talk about a little bit more in the days to come. So the nervous system is basically a complex network of nerves and cells that carry messages and information to and from the brain and spinal cord to all parts of the body. So the nervous system is basically responsible for the integration and control of all the body's functions. So it's a very essential part of who we are. There are 2 major divisions to the nervous system. There is the central nervous system and the peripheral nervous system. And you guys a lot of you probably already know this. So like I said, it's just pretty basic. So, okay, here's looking at the divisions of the nervous system. We're going to start by talking about the central and peripheral nervous system. Okay, the central nervous system is basically the brain and the spinal cord. So it's all the nervous tissue that is enclosed by bone. The brain and the spinal cord allow the body to detect and respond to both internal and external stimuli. Internal would be different functioning of the organs and things like that, digestion, and external, of course, is things that go around in our surrounding environment. Okay, the peripheral nervous system is all the nervous tissue that is not enclosed in bone. So this includes the tissue that transmits information to and from the central nervous system. So you have 12 pairs of cranial nerves and 31 pairs of spinal nerves. And you can see from the picture, the cranial nerves are up around the head and the spinal nerves come out from the spinal cord and out to different parts of the body. There are 2 main types of cells in the peripheral nervous system. There is the afferent cells, which are the sensory cells. These cells carry information from the outside to the nervous system. And the efferent or neurons are basically the motor neurons. So these are the signals that the central nervous system sends out to the body. So it's sort of like the efferent goes out, the afferent comes in. Okay, and the peripheral nervous system is then broken down further, like into the autonomic and the somatic nervous system. The autonomic nervous system controls the internal activities of the organs and the glands, while the somatic nervous system controls the external actions of the skin and the muscles. We're going to talk a little bit more about these. So again, the somatic nervous system is associated with voluntary control of the body movements. So this includes the muscles and all the movements that we make, whereas the autonomic nervous system is associated with basically with breathing, with your heart rate. These are the things that you don't have to attend to but your brain is sort of attending to them for you. You don't have to attend to them consciously. Your brain is keeping these in check for you as we go throughout the day. The autonomic nervous system can then be further broken down into the sympathetic division and the parasympathetic division. Those are the two main ones, and here they put the enteric division as the gut. But we're going to talk more about the sympathetic and the parasympathetic. So back to the somatic nervous system. This is the one that has our voluntary control of the motor system. So again, there's the sensory neurons that go in, and the motor neurons that go out. So here's just an example of what would happen if you touch a hot object. Okay, so basically your sensory neurons convey information from the receptors for pain, temperature, and things like that. So if you touch something hot, there's an automatic pulse that's sent up into your central nervous system, and immediately it activates your motor neurons to pull your finger back. And you know this happens very, very quickly. If it didn't, we probably would be in trouble. And again, the motor neurons return these impulses, and they're the ones that help us to react to our environment. The autonomic nervous system, again, is concerned with the involuntary regulation of both smooth muscle, cardiac muscle, and glands. So smooth muscle are the muscles that contract our tissues. So like bladder, the heart, and things like that. They're not the skeletal muscles. They're muscles inside us that do different things for our organs. And the autonomic nervous system, again, consists of the sympathetic and the parasympathetic. The sympathetic is the arousing part of our nervous system. So it's sort of the fight or flight type of response. If you're put into a situation where you need to react, your sympathetic nervous system kicks in. And what it does, basically, it works to constrict the skin vessels and blood vessels throughout the body, and it starts sending more blood to the heart and to the lungs. So your heart rate increases, and the blood vessels to your skeletal muscles dilate, so you can send more blood to your skeletal muscles, so you can react quickly to whatever situation you're put in. The parasympathetic is basically the calming part of the autonomic nervous system. So this is the opposite of the sympathetic. So when we're relaxed, the parasympathetic nervous system kicks in. It allows the heart rate to decrease, and it allows the body to return to sort of a normal, calm state. So these are two of the different ways that these sympathetic and parasympathetic nervous system works. So on the right there, the sympathetic nervous system, if you're put into a situation where you need to react, it will dilate your pupils. It inhibits the flow of saliva. So anything that has to do with digestion basically slows down when you activate the sympathetic nervous system, because you need that blood and you need that energy to areas of the body that are reacting to something dangerous or something that's getting you sort of stirred up. It accelerates the heartbeat, it dilates the bronchi so we can have more air and more oxygen, inhibits, like I said, the digestion, inhibits bladder contraction. Whereas the parasympathetic nervous system, it's more of you eat a big meal, you want to sit down and relax. It kicks in, it allows you to do peristalsis, which is the contraction of your digestive muscles, slows the heartbeat down, constricts the bronchi. So again, sort of the relaxed state. So while we're busy doing all kinds of other things and attending to things, our autonomic nervous system is always under this regulation of these two balance mechanisms. Okay, so now we're going to talk about the cells of the nervous system. We have neurons, which is the most probably familiar cells to people of the nervous system. These are the cells that conduct the electrical signals for us. and we have glial cells. And these glial cells make up different parts of the nervous system. And it used to be thought that these glial cells basically were for support of the neurons. And you'll see, I'll give you some examples of things that these glial cells do. But people are paying more attention to these glial cells now and doing more research on them and trying to determine their importance to a greater extent these days. So they're being less ignored, basically. Okay, the neurons have two main parts. The dendrites, which is the part that receives information from other neurons. So this is the intake part of the neuron. And the axons. So the axons are what sends the information along to another neuron or to different parts of the body. And these axons can extend between being a few millimeters long to even up to a meter long. And like I said, they send the signals out. Okay, so here's some different types of glial cells. Like I mentioned, the glial cells were previously just thought to be sort of the supportive cells, but they are the most abundant types of cells in the nervous system. And they don't conduct electrical signals, but they do have several other functions. So we have these oligodendritic cells and Schwann cells. And I mentioned the fatty tissue in the brain. These are the cells that produce this fatty tissue to form myelin around the neurons, and we're going to talk about that a little more, the myelin later. This myelin helps us transmit signals in a quick manner throughout the body. So the microglial cells, they scavenge and remove dead cells. So as we go back, we can take a look here. The Schwann cells are the myelin producing cells. The microglia cells are sort of the scavengers, so they go and clean up the dead cells and tissue. Astrocytes and satellite cells provide nutrients and support for neurons in central and the peripheral nervous system. And the radial cells provide sort of a bridge for developing neurons. So they provide the infrastructure sort of for the neurons to go and take the paths that they do. The ependymal cells are, they line fluid-filled ventricles of the brain, and these are the cells that produce the spinal fluid that bathes the brain and keeps it in this sort of moist environment. So you can see here the ependymal cells and the astrocytes that also provide some of the structure for the neurons to develop. Okay, so neuron communication has two main types. There's communication within a cell, and this is called neuroconduction, and this is how the signal goes from basically from the dendrites down the axon throughout the cell. And then there's transmission between cells, and this is called intercellular, whereas within cells is called intracellular. So between cells is called neurotransmission. And this is how one cell talks to the next and passes along information or signals throughout the body. Okay, neuroconduction is electrical in nature. So it acts much like an electrical signal in a wire, but instead of using electrons that pass through a wire, it uses ions that move in and out of the cells. So nerves at rest, meaning there's no stimulation to them, they're just hanging out waiting for something to happen, have a negative net charge inside them compared to the outside of the cell, and this negative resting voltage is called their resting potential. So they're just hanging out waiting for something to happen, and along comes an action potential. This is their signal that they're going to get ready to work and pass information along. So when a signal is received, there's ion channels in the cell membrane of the neuron that open up. And when they open up, charged particles flow into the inside of the cell. And I'm going to show you a video of this and a little bit more about this. So I know this is a lot of information pretty quickly, so we're going to slow down. I'm just going to give you the basic overview of what happens first. Okay, so these charged particles are flowing into the cell, and as they do so, the electrical charge of the inside of the cell changes compared to the outside of the cell. So it becomes more positive. Remember, at the resting state, it's negative. These positive ions are flowing in, and the cell potential is becoming more and more positive. And once it reaches a certain threshold, that's it. It's going to fire. There's no going back. So it fires. It sends this electrical signal. The signal is propagated along the axon to the next group of cells on the membrane, and the cells in that certain region repolarize and return to the resting state. So this is basically, the signal is moving along the axon with this, the cells coming in and out and changing the electrical potential as it goes along. So the action potential in terms of our membrane voltage looks something like this, okay? So at the very beginning, we're at a negative resting potential. So down there at the left would just be the cell at the top with the channels closed, just sort of waiting for something to happen. Then an action potential comes along and these sodium channels start opening. So sodium and potassium are the main two ions that cause this process to happen. So when it receives this action potential, the channels start to open and sodium flows into the cell. This is making our cell membrane go from negative to more and more positive. And interestingly, the calcium channels open and the calcium begins to leave the cell, but the influx of sodium is higher compared to the outflux of potassium. So the membrane potential becomes more and more positive. And once it reaches this threshold of excitation, like I mentioned, then the cell will fire. So what happens is the sodium channels keep coming in until it reaches a certain point where they close and no more are going to enter. And this is the cell firing, and then it returns back down to a resting state after the channels close. Potassium continues to leave the cells, causing the membrane potential to return to a negative resting potential down here at the bottom. And it goes through this refractory period after it fires, just sort of like it's returning to normal state. So when it's in this refractory period, it can't fire again till it gets back to its normal resting potential state again. Is that making sense? Is everybody following okay? Okay, so we're going to watch a little video of it. I know I'm driving it into to the ground, but
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in.
I think I might have gotten that backwards.
While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge.
Okay, this is resting.
The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
Okay, so you can see there the example of how that propagates and how these channel pumps work to change the electricity and move the signal down the axon of the cell. So you might be wondering how quickly this can occur, because we do need, like we said, our nervous system to be fast. If we stick our finger in a fire, we want to be able to pull it out pretty quickly. So we don't want to be waiting all day for our channels to open up and close and say, oh, okay, you know, I have a nice burn now. So it does happen very quickly. The velocity of this action potential and conduction can vary from 0.25 meters per second to 100 meters per second. And generally, the bigger your axon fiber is, the faster that this conduction can go. But to increase the velocity of this nerve conduction, we have what's called myelin informed around the nerve cells. And I've mentioned this a few times, that myelin is that fatty tissue that wraps around the axons, and it acts sort of like an insulation to help this action potential move along more quickly. And the myelin, again, is produced by these glial cells that we saw before. They're actually called Schwann cells that secrete these and wrap around the axons, so that the action potential, instead of traveling like we saw all the way along the nerve, can actually jump from node to node where these where this myelin has a little break in its tissue. So let me see if I can show this a little bit better in this slide. So this myelin here is wrapped around the axon, and the places that have little breaks in the myelin are called the nodes of Ranvier. And so instead of having to propagate the nerve conduction all the way down the axon, instead it's jumping from node to node, and this helps to increase the velocity of the nerve conduction. These myelin sheaths are what actually give the brain the white color that we call white matter. So the gray matter is the neuron tissue, and the white matter is full of these connections between these neurons and wrapped in the myelin for very fast conduction. And this is actually some brain images from one of the studies that we do, and you can actually take the slice of brain image and break it down into gray matter and white matter. And you can see really how much white matter you really have inside your brain on the right-hand pictures there. The left-hand pictures are the gray matter, which we're going to talk a little bit more about when we talk about neuroanatomy. But it's this white matter that really allows for really rapid communication between the cells. Okay, so I said between the cells, actually I meant within the cells, because between the cells we have what's called neurotransmission. And neurotransmission is the communication between two different neurons. And the space between two different neurons is called the synapse. And at the synapse, the electrical signal cannot jump from one cell to the next. So there's another mechanism that's used to send the signal across the synapse. So at the end of each axon, which are these long strings that come from the cells, are these knob-like protrusions which hold synaptic vesicles that hold what is called neurotransmitters. And you may have heard of these neurotransmitters before. When a signal is received, this electrical signal, these neurotransmitters are released into the space between the cells. And again, this space is called the synapse. They diffuse across the synapse and they bind to receptors on the next cell. And remember, we said that what brings the signal in is these dendrites. So the axons come down, the space between is the synapse, and they send the signal to the dendrites on the other side of the synapse. These dendrites have the receptors that bind the neurotransmitters, and this causes them to start to send the action potential down their own cell body. So, and propagate the signal along to the next cell, and again, then this neurotransmission occurs at the next synapse. So here's a picture basically on the left of the terminal of an axon. And this again is this synaptic button or button, and it's full of these neurotransmitters, you can see. And when the cell receives a signal, it dumps these neurotransmitters into the synapse, and it can bind on the other side, causing the next cell to fire. And on the right is, you can see the microscopic view of it. The synaptic cleft there is labeled. I'm afraid if I walk away, you won't hear me. But here's the synapse here, and you can see these vesicles coming to fire to the next cell. Okay, so we're going to watch a quick video of this, because I think it can explain it probably better than I do.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, so that was just a brief little illustration of synaptic transmission. So these are how the cells communicate between each other. Okay, so you saw that the neurotransmitters are oftentimes repackaged into the cell and recycled, which is actually a pretty clever little mechanism that the body uses to keep from making more. So again, these neurotransmitters are these endogenous chemicals that transmit signals from a neuron to a target cell across the synapse. And they're packaged in these little vesicles, as you saw, and they're released into the synaptic cleft, where they bind to the receptors on the other side of the synapse. Now, these neurotransmitters can either excite or inhibit a process. So they can come and bind, and they can be sending a message for the cell to fire or for the cell actually not to fire and stop the process. So they don't always have to be excitatory neurons. After the transmission, the neurotransmitter can be recycled, degraded, or diffuse away. So you might ask, what are these neurotransmitters? What types of neurotransmitters do we have? And I think you probably heard some of these words before. The type of neurotransmitter depends on what type of system the brain is using. So here's some examples. Like acetylcholine is a big transmitter that is involved in muscle contraction. So this is involved when muscle signals are sent through the nerves into the muscles. And acetylcholine is also involved in wakefulness, attention, anger, sex, and other things. So it's an excitatory type of neuron that propagates sort of a muscle or a movement signal. Dopamine is another type of neurotransmitter that's involved in controlling movement and posture. GABA is a neurotransmitter that's widely distributed in the brain for motor control, vision, and anxiety. Glutamate is also an excitatory neurotransmitter that's often associated with learning and memory. Neuroepinephrine is a neurotransmitter that's helped in attentiveness, emotions, sleeping, learning. And serotonin, you might have heard of, is a big neurotransmitter that regulates body temperature, sleep, mood, and appetite. And in the past several years, we've heard a lot about serotonin, at least in the U.S., because we have a lot of people now on these called SSRIs that we're going to talk about in just a minute. So one strategy for treating or for understanding what these neurotransmitters do is to look at what happens if you lack these neurotransmitters, okay? So if you lack acetylcholine, this is often associated with Alzheimer's disease. Parkinson's disease is from a loss of the dopamine neurotransmitter. So, in Parkinson's disease, people often shake. This is the loss of these fine motor skills that dopamine helps to facilitate in our brain and throughout our body. Mood disorders are often associated with norepinephrine, and mood disorders are often also associated with serotonin. Okay, so as I mentioned, these SSRIs, these are called selective serotonin reuptake inhibitors. And one prime example of this is Prozac. So one theory that about depression is that it's caused by low levels of serotonin in the brain. So if you go and tell your doctor you're depressed, he might give you Prozac or some kind of SSRI. Prozac was the initial original one, and now there are all types of SSRIs. So they're actually medications that block the reuptake of serotonin. And serotonin, like I said, is thought to be associated with mood and different things like that. So what happens is these medications get into these little holes where the transmitter would be taken back into the cell after it fires and goes across the synapse, and it blocks the neurotransmitter from being taken back up by the cell that released it. So it stays in the synapse longer and increases the activation of the receptors on the other side. And this is, I guess it's proven to treat depression in many cases. Okay. So, in summary of this very brief introduction to the nervous system, the nervous system is basically a complex network of cells and connections throughout the body. The nervous system allows for communication at a voluntary and an involuntary level. So, at the microscopic level, communication involves nerve conduction and nerve transmission. So again, these are the conduction of the signal along a cell and across between cells, and these signals are both electrically and chemically propagated. And so as you can get from this little summary, the nervous system is a very, very essential part of our body. It keeps all of our organs in check and in regulation. And it's constantly working in our background to keep our body flowing like it should be flowing, and to make sure we, don't stop breathing or don't stop digesting and things like that. So it's a very essential part, and it's probably my favorite organ system. Right, that's all I have for today. And I'll probably stop there instead of going into our next lecture, since we don't have a lot more time. But if anybody has any questions, I'd be happy to answer. Or if you want to ask me, privately after or in front of the class, feel free. Okay. Yes. So the question was, do all neurons have all types of neurotransmitters, or do they only have certain neurotransmitters? The answer is they only have certain neurotransmitters depending on what kind of neuron where they're involved and what they're actually doing in the body. So for example, dopamine, where we'll talk about a little bit more later, is involved a lot in the reward centers and in the midbrain. GABA is a transmitter that's used a lot in the frontal lobe of the brain, and GABA is sort of helps to inhibit processes and behaviors. For example, Phineas Gage, and he had a lack of control. GABA is one of the neurotransmitters that helps with that. So it really depends on what that function of that neuron is, what the communication function is, and where it's located in the brain. Good question. Sure, of course. Please. So the question was how the glial cells, there's some types that help degrade tissue and get rid of dead cells. And I'm not so specifically, but I know that other cells that in the body that do this, they actually come and they will sort of just eat that tissue and degrade it down. So it's actually pretty interesting. Yeah. In fact, our immune system also has sort of cells that do that, but they go after the bad cells and the viruses and things, and it's called endocytosis. They take that bad tissue into the cell and they sort of surround it, and then they break it down. Okay, thank you very much for your attention today, and I hope you guys found that useful, and we will get back together tomorrow. Tomorrow at 9:30.